Hi everyone, my name is Tong Wang from Shanghai Tech University. Today I'm going to introduce a mini review that we recently published on BioEssays. The title of it is about the axonal radial contractility, structural basis underlying new form of neuroplasticity. As we know that the neuron is the longest cell in the human body, and the majority of its length is occupied by its axon, which can extend up to 1 meter from its soma to its terminal in human bodies. And this one is not only the cables for electrical signal transmission, but also the important tunnels for material transport in the form of axonal cargo trafficking. In our previous works, we have established a system using microfluidic devices to study the long-range axonal trafficking. And from this live imaging data, we have found that it seems that different sized cargoes has different trafficking speed. The big ones seems to be always stocked easily than the smaller ones in axon trafficking. And based on this live imaging data, we have done some statistic quantifications, and we confirmed that yes, the big ones actually moves slower than the small ones in axon trafficking. And the most likely reason we think is because it receives radial tension from the axon walls that is very thin and restricting its trafficking. And this hypothesis was actually supported by our data using the TEM, showing that the axon was actually expanded when there is cargoes inside. And this kind of extension is very obvious. So this was further supported by the time-lapse super-resolution microscope, showing that the axon walls actually expanded when the big cargo passing through. And so therefore, we have formed our hypothesis. We think that the radial contractility of the axon is a new form of structural plasticity in axon that allows the large cargoes to pass through smoothly in the long-range trafficking. And we further explored the structural basis for this new form of structural plasticity. And we found that the actin rings peristaltically distributed along the axon is its structural basis. That these rings can be expand and reversibly retract upon the passage of large cargoes. Therefore, we summarize our findings here as that the axon can be radially contractible, and the structural basis is those periodic actomyosin rings. The following question is that how these rings can be easily contracted. So we have two hypotheses explaining that. One is that it is composed of very small actin fibers that can, when the tension comes, it can be extended apart from each other, and as a whole, the ring extended in the diameter of the ring is expanded. We call this closed ring hypothesis. The other possibility is that the rings is actually composed of two long actin fibers, but it has an opening. It is an openable ring instead of a closed ring. Therefore, when tension comes, it can be opened very easily and reversibly to allow the passage of big cargoes, and then it can be closed afterwards. So we all know that now actin is not an inert fixed diameter cable. It is plastic and has a radial contractility. And this is the new form of structural plasticity for neuron. Thank you.